right. First of all, thank you very much for inviting me to the Longevity Conference. Also, big shout out to the whole team for organizing this amazing conference. It's really enjoyable for me. Listening to the talks yesterday and also especially this morning really confirms my notion that I'm suffering from imposter syndrome. I'm professor of pathology, I'm not a pathologist. The advantage is to be in the pathology department is that I can do anything I want. I don't have to justify working on this organ system or that organ system. I talk about immunology often enough. I have no idea about immunology. I'm a trained organic chemist. And thirdly, I'm here at the Longevity Conference, and I'm not studying aging. Actually, I'm looking at the other end of the age spectrum. I'm at Boston Children's Hospital, and we are particularly interested in what's happening after birth, in the first hours, days, weeks, months of life. So at the same time, actually, I think we can learn from each other. After all, longevity works towards one defined endpoint, death, mortality. We are working from one defined starting point, birth. So that's one aspect. The other aspect is also, as it was discussed yesterday, that actually very often a lot of the longevity studies, they start in early adulthood. But I personally think that childhood is incredibly important because it's going to have a major impact on what's happening basically throughout your lifespan. Anyway, that was a long preamble. Now let's get started. And after all those high-flying talks this morning, I will bring you back down to reality here, or down to the ground a little bit. I was very happy to hear from David that proteomics was the best omics for his molecular clock. So I'm very proud. So thank you very much for the kind of nice introduction. So, however, when you talk about proteomics to other people, what do people associate with proteomics? And they associate normally slow, it's expensive, and it overpromised and underperformed. And as much as I like proteomics, I do have to agree that used to be proteomics. However, that is the old proteomics. Over the last 5-6 years, there's something, what I would say, there's a new proteomics. We are dealing with unprecedented sensitivity. So we can do now single-cell proteomics. We can identify 500 to 2,500, 3,500 proteins from a single cell on a mass spec. And we have unprecedented speed and robustness. So that gives us really unprecedented throughput, something that we had never before. And if I just look back at basically how my lab evolved, we started out with single samples. We're proud when we had 5 or whatever. Then eventually we moved to the tens, hundreds, and we were really proud when we had the first study with 1,000 samples. We were really whoa, we are really out there. It's really great. And what happened in 2020? COVID fell out of the sky. And that was in the year also, or the time, the NIH contacted me and asked me, hey, Hanno, we want to do this COVID study. Can you analyze 10,000 samples for us? So I swallowed hard, and I realized I had no idea what they were asking because nobody had ever done 10,000 samples in proteomics. And so I decided, well, I might as well say yes, because, if you pull it off, great, and if you don't pull it off, I'm just as bad as everybody else. So I thought I couldn't lose. So, and 10,000 samples are actually a lot of samples. So we had to learn. I should also mention, because it was COVID, was a special situation, we had 3 months to increase our throughput by factor 10 and kind of tell them, okay, this is a method. We had to lock it in because it was a clinical trial also. So we didn't, it was a very interesting period. But ultimately, we pulled it off. So when it comes to plasma proteomics, people kind of often liken it to an iceberg. You see a little bit that comes out of the water, but that's actually just a minor part of the iceberg. The majority is underneath the surface. And so people differentiate that between what they call the classical plasma proteome, the tissue leakage plasma proteome, which is very often really what kind of people want to see when it comes to biomarkers for cancer and so on. And then there's also the cytokines and chemokines. And as much as I love proteomics and mass spec, I have to acknowledge the fact that mass spec is not the method of choice when it comes to cytokines, chemokines. You're much better off using Luminex, Olink, Alamar, you call them, basically good antibody-based methods. But we were interested in coming up with a kind of a better method to analyze the tissue leakage proteome. Because, as I said, that's where people think they're the proteins of interest. So until then, basically until 2020, most of the people that were trying to get to this used antibody-based methods. And again, antibodies are great molecules, but it's really nothing you can do really that well on a large scale. It's really slow, it's expensive, it lacks robustness. We know that small changes at pH, small changes in salt concentration can affect affinity and specificity. So that was really nothing we could do, especially not on 10,000 samples. So there was an author in my lab, a French postdoc at that time. He dug out a paper from 1948, so good old protein biochemistry. It actually turned out that perchloric acid does a fabulous job depleting or precipitating the most abundant proteins in plasma. Admittedly, we still don't fully understand the protein biochemistry. All I can say, it does work. So that was the workflow that we had. And basically, let me go back. So we have here the acid precipitation step, and then we went for the supernatant. And then everything was put on the EvoSep system connected to a Bruker system, which is in my hands kind of most robust LC-MS system out there right now. And as I said, it does work. I want to show that here with this plot that basically show the rank of the protein in neat plasma for the top 10 proteins, and then after the depletion, how the rank changes. And you can see all of those ranks increase, meaning they're getting depleted. And for instance, here albumin, even though it looks at rank 35 from rank 1, it means that basically you can do the math. It turns out that 98 to 99% of albumin is depleted in this method. So it really kind of opens up the door to take a much closer look at what's underneath the surface of, or kind of taking off the top of the iceberg so that the iceberg comes up and more things come out of the water. It's really cheap, the reagent. We're talking about 3 US cents or translated into 3 rupees per sample for the reagent. And yes, there are other methods out there, but you easily talk about $100 per sample, which translates into close to 10,000 rupees per sample. So if you really want to do it on a shoestring budget and want to do thousands of samples, this is kind of certainly a good argument to go with this perchloric acid method. Then in our hands, it's incredibly robust. What you see here is a PCA analysis. Each dot is 1 sample. You can start counting. I can help you. There are 3,199 dots. They were analyzed or processed and analyzed in 3 batches over a 10-month time period. And I hope you agree with me that there's not much of a batch effect. So we were really proud of this, actually. So and also just to show that it really helped in sensitivity, or kind of that we could really dig deep into the plasma proteome. This was one of the few studies that actually allowed to detect in discovery mode SARS-CoV-2 derived proteins in plasma. And always keep in mind that SARS-CoV-2 was not a bloodborne pathogen. That's why they didn't do blood tests for COVID testing, but basically nasal swabs and so on. But we could readily detect here this nucleoprotein with not only 1 peptide or whatever. We basically had more than 20% sequence coverage. So this was a rock solid hit. So all looked good. And all of that also, as I said, on an unprecedented throughput of 100 samples a day on a single instrument. The instrument is a bottleneck in many ways. On the one hand, it's okay because it's the most expensive part of the workflow. You always want to make sure the bottleneck is the most expensive part. At the same time, it also means that it's scalable. Basically, more instruments means more throughput. So what do we gain actually when we go to larger cohorts? And again, proteomics used to tinker around with 10 and 10 samples or 20 and 20, whereas now in my lab, we are trying to do pilot studies with 500 samples. You get just so much more information out of 500 samples than you get out of 50 or 20. So there are kind of just a couple of examples of what you get out of larger cohorts. Let's start with here deeper insights and more accurate and robust information. This was, again, the large-scale COVID study that triggered basically the whole development, the whole method development. And the study design was basically we had 5 trajectory groups for the patients. All patients were hospitalized COVID patients, and TG1 were basically they were still sick enough to be hospitalized, but then well enough to be sent home within 3 days. And trajectory group 5 were basically people who died within 28 days in the hospital. And then on top of that, we had up to 6 samples per patient during the hospitalization. So all of that, again, even though we planned for 10,000, turned out to be only 4,500 samples. And these are the people who pulled off the feed at that time. Kind of was interesting. Everybody dropped his or her project and really coalesced around this one mission. We want to have this pipeline, and we want to analyze these thousands of samples. Well, I guess at the beginning of COVID, there wasn't really not much else to do anyway, apart from working. So it was a kind of, it worked out for everybody, let's put it that way. So again, the nuclear protein wasn't also interesting to notice that actually it didn't really matter how sick you were. You basically almost had the same level of this nuclear protein in the plasma. However, what did change was the frequency observation. Basically, well, 40% of the very sick people had this particular protein, or the viral protein, in the plasma. Only 4% of the least sick ones. So this kind of a level of information, again, just so kind of, it's almost in the level of fun facts, but it's certainly interesting also. That it's not always about levels, but it's about also observability of frequency. Then you get plenty of information, all those heat maps. What I'm showing here is just a heat map for the samples at time of admission to the hospital. So we didn't even, at this moment, didn't even bother to look at the different time points. And if you're interested in the whole study, that was published last year in Science Advances, so happy reading. But then we had the luxury of multiple samples or longitudinal sampling, and I think that's really also important. After all, that was also kind of discussed a little bit this morning that, you know, yes, you can do a CT scan or MRI scan, and then when you see a cyst or lesion, you can do surgery, but as we heard, it might not be a good idea because we might overdiagnose. And I think important is really that you want to do multiple testing or kind of multiple time points. Because, you know, if you do a CT scan and you see a cyst, okay, sure, you take it out or not. One solution would be wait a year, do it another year. If it doesn't grow within that year, then you might as well leave it in because it doesn't look. It's certainly something to keep on the radar screen, but you can ignore it at that moment. And that's also here what we had now, for instance, here with the different time points. Here, for instance, when we looked at proteins associated with degradation of extracellular matrix, that we could clearly see that depending on the severity, we had different levels. And was also interesting, even though we differentiate between trajectory group 2 and 3, they really always kind of went hand in hand. But basically, the more severe you were, the higher the abundance of those particular proteins. Another example was here, for instance, for NET-associated proteins. Oh boy, I really have to hurry up. So here, for instance, TG4 and 5 started out at the same level, but basically then the people who survived managed to keep it under control, whereas the people who didn't survive, there the net proteins just kept increasing. So just as an example of importance of longitudinal several time measurements, because you get so much more information at that moment. All right. Then we had, the definition of TG5 was death within 28 days. That means that anybody who died, for instance, on day 29 or 30 were automatically in trajectory group 4. That gave us a possibility to separate actually TG4 and TG4 survivors and fatalities. And actually turned out that the TG4 fatalities was definitely much more similar to the TG5 group, meaning that there was something molecularly clearly indicating the basically upcoming death. And for instance, here trajectory, this histone 1,5 protein was one of the death markers that we had. And also following up on one of the talks this morning, actually this H1,5 protein turned out to be also one of the death markers for Ebola, reminding us it's normally not the virus that kills us, but it's really our own immune system that kills us. And we came up then with this death panel, and we could basically prognosticate with an accuracy around 80–85% whether somebody would die or not at time of admission. All right, let's hurry up here. The next one was more highly resolved measurement series, and you get very interesting, can get very interesting information out of that. That is here, this particular study. It's an EPIC study, we called it. It was actually a vaccination study for newborns. It was one of the NIH or NID-funded Human Immunology Project Consortia projects. And the proteomics team is shown here, Benoit, Tour, and Kinga. We piloted it. That was the study design here. 30 kids gave a blood sample. And again, we're talking about newborns, day of life 0. And then gave a second one. A third gave a second blood sample on day of life 1. Another third on day of life 3. And a last third on day of life 7. We did a lot of proteomics, metabolomics, transcriptomics, and so on all these samples. And then we looked at what's changing during the first week of life. And it was, even for me, impressive to see that even within day of life 0 to day of life 1, quite a bit of is changing. If you look at it from an immunological perspective, it makes sense. We can discuss it during coffee break or during lunch if you want to know more about the immune system of neonates. And you can also read up on this in this paper. But the problem is when you only compare day of life 0, day of life 1, all you can do is basically say something goes up, something goes down, or something is stable. So now, however, the real pilot or the real EPIC HIP-C study with large cohort, we actually had 720 newborns. So here, 720 newborns, a third of them gave a second blood sample. That actually afforded us with the opportunity to split them up into 2-hour windows. So instead of looking what's happening between day of life 1 and day of life 0 and day of life 1, we could now follow with hourly resolution what's actually happening in the plasma of newborns immediately after birth. And here, one example, for instance, IL-8. What you see here are the measurements between day of life 0 and day of life 1. Statistically, not much of a difference. You probably would say, it's going down. Didn't quite make sense that it goes down, but nevertheless, those are the measurements. But now filling in the gap actually turns out to have quite a complex dynamics that you would never get if you just take 2 measurements. Now we have 24 measurements. Yeah, I know. People, if you're hungry, go out and eat, or you stay here. And so, for instance, when we look at IL-8 and IL-10, was actually really interesting to see how the one pro-inflammatory and the anti-inflammatory cytokine first really showed totally different behavior, and then eventually they show change that are in parallel. And also here, for instance, when we look at the proteins that go up, we actually had very different trajectories. We could see a very clear ordering of some proteins that immediately go up and reach basically half maximum in less than 6 hours, after more than 6 hours, 12 hours. And then we had also some delayed proteins and some other proteins that were still at the exponential growth phase. And then we had something similar for the proteins that went down. And that's a level of detail you only get with higher time resolution, and higher time resolution requires many more samples than what you would need if you just want to look at something, whether something goes up and down. And you can clearly see that it's a difference whether something goes up or whether you get this dynamic information out of high-resolution time-course experiments. Since molecular clocks are here en vogue, we also decided to use a elastic net-based model using the proteomics data. And actually, it did work quite well. And I just would like to highlight here really our x-axis. This is not weeks, months, or years of life. These are hours of life that we try to model with our molecular clock. And what was then also interesting, actually, when you then correlate for each time point, you get a protein profile, you correlate all the protein profiles, you actually get critical phases immediately after birth. And one is after 16 hours, and one is after 40 hours. We don't fully understand from physiological level what's going on, but it's robust. We saw it for the proteins, we saw it for the chemokines and cytokines. So it's something we're working on with the pediatrician neonatologist to understand what's going on. And since I heard already 3 bells, let's just move on here. I skip all that. Sorry about that, guys. And conclusions. Recent advances in plasma proteomics really allows us now to take on studies with thousands of samples. We can really do population-based studies. Our cost-efficient plasma proteomics, our depletion protocol also lends itself to applications in larger cohorts. I hope I convinced you that large cohorts gives you much more useful information and unique insights than other kind of smaller cohorts would do. And then also, I should mention that even though I only showed you data for plasma, the whole pipeline that we have also works for many other body fluids, CSF, urine, saliva and oral secretion, tracheal aspirates, and dry blood spots. Because I should mention that since we work in preemies also, we normally don't get blood samples for research purposes, that we have to work with other body fluids. We mostly look at urine, oral secretion, and dry blood spots because those are more readily collected in the NICU. And basically, the old proteomics, instead of slow, expensive, over-promise, under-promise, the new proteomics is fast, it's actually cost-efficient, and it's sample-efficient and ready to take on large cohorts. And people get nervous here. You can see I would like to thank the people who pulled off the high-throughput pipeline again, then the Steen Labs. My wife has also a lab in the neurology department. I guess that I'm the junior partner at home and at work. And all the funders who keep the lights on in the lab. And with that, I would like to thank you for your attention. Happy to answer questions.